Today I'm going to introduce a scientist called David Liu. This is about his journey in science. So starting from childhood, Liu has developed an interest in science by playing with insects and also making small explosives by using items from his home. He later received education from Harvard University and the University of California, Berkeley. He first focused on the synthetic chemistry and biochemistry, and then focused on protein evolution techniques in his graduate school. He was a researcher in different research institutes and also joined funding to different technology companies such as Antus Medicine, which focused on CRISPR technology treatments. He has a lot of interesting hobbies, such as he's really good at playing blackjack. He even got barred from all MGM grant facilities because he won too much money at the table and he was so good at it. And he was so obsessed with the math in this game. Also, I like to spend time on making radio-controlled airplanes, which can fly as slow as 1 or 2 miles per hour. And this is a picture of him and his airplane. Apart from those, he also liked to make some small inventions and creating arts from different materials. And the reason why I admire him, there's a lot of reasons, but the most important thing is that he showed great resistance when facing doubts from others. Even when others said his idea is not going to work, he still insists on doing what he was doing, and finally he got succeed. And I really want to be such kind of person. And he was in the field of molecular biology, and their group mainly focused on integrating chemistry, like those small organic molecules, into biology to create new therapies in the genetic diseases field. And these are three core areas of his studies, and I'm going to explain more in later slides. And this is a research article published in Nature from their lab. And basically, it just talks about a new technology, a single prime editing system, which can be potentially treated to multiple genetic diseases instead of just one. And in this figure, they investigate the effect of the technology that suppress tRNA on the animal models of mass. And they just use the prime editing technology and they inject the AAV9 vector to deliver the suppressed tRNAs to the mice with a genetic disease. And then after several days, they analyze the samples from different organs to record the editing efficiency and the enzyme activities compared with the control group of the wild type mice without the prime editing. And because they found a significant difference according to a p-value in the enzyme activities between the experimental and control group, they reached the conclusion that this technology, the prime editing with the suppressed tRNAs can actually restore enzyme functions, which is usually suppressed in which cause genetic diseases. So actually, this technology can be served as a potential treatment. And in the past, he and his team developed different technologies in genetic editing, such as base and prime editing, and also they developed DNA template synthesis and also the phage-assisted continuous evolution. In the future, they want to focus on increase the level of precision in the genome editing technology, and also they want to make the personalized treatment more accessible and increase the scale of patients. And this is a list of reference that I cited in this presentation. So in this presentation, I introduced David Liu's life experience, hobbies, and his research focus. Thank you for listening.